Welcome to the Musella Foundation's Brain Tumor Webinar Seminar series. Sorry. Tonight's special guest speaker is Santosh Kesari. He's a director of and chair of Neuro-Oncology, Department of Translational Neurosciences and Neurotherapeutics at Pacific Neuroscience Institute. Take it away.
Well, thank you, Al, for inviting me to come to give this talk on a Sunday afternoon. Happy Father's Day to everyone out there.
Happy Father's Day.
Yeah, happy Father's Day. So I've never done it live like this before, but I'm happy to take questions as we talk or at the end, I guess. What I thought I would do is really just spend some time talking about immunotherapy for brain tumors. This immunotherapy has been around for many, many decades, and it hasn't really shown much progress until recently, in the last decade or so, in other cancers. And now we're trying to apply it for various types of brain tumors with mixed results. It is still early days, and I think this is a very promising approach for brain tumors, and we just need to understand a little bit more how to use immunotherapy, what types of immunotherapy, and timing of immunotherapy, and what combinations may be best along with the standard of care treatments that we're doing currently. So as I think everyone knows, of brain cancers, glioblastoma is the most malignant of brain tumors. And what we try to do, the standard of care, which was established in 2005, is really to do the maximal surgical resection, safely, get as much tumor out as possible, and followed by radiation and temozolomide chemotherapy for about 6 weeks, and then followed by adjuvant chemotherapy. And as you can see on this slide, the median survival is significantly improved, as well as the 2-year and 5-year survival of this regimen. Since then, we have an additional standard of care where we add Optune with radiation and temozolomide, and that also affords another 2 months of additional survival. Median survival and 5-year survival is also improved. So those are the standards of care. And even at 5 years, though, we're still in the about 10% range of survival for most of our patients. And so clearly, even with these new standards of care, there's still a tremendous opportunity and need to improve survival. And that's what all the clinical trials are focused on right now, is can we do even better? And so one of the things that's clear, the reason we made some progress, but we need to make faster progress, and it really is due to these 5 things. The tumors that develop in our patients, they're all heterogeneous. Every person's tumor is different. They look differently on pathology. They have different genetics. Each tumor, when you profile them, have different mutations, different gene amplifications, different deletions. So that contributes tremendously to the difficulty in identifying one drug for all of our patients, and that we really need to personalize treatment for each patient based on the genetic profile of the tumor. And that's becoming increasingly clear, that we need to do a better job of understanding this, what we call heterogeneity, and then applying that heterogeneity in clinical trials so that we can do smarter, smaller trials with targeted drugs to see if they work or not. The other thing that's clear is many of our drugs don't get into the brain tumor. And this is in part due to the blood-brain barrier, but there's also some other variables, including delivery and angiogenesis that is inefficient, that doesn't allow all the drug to go to all the right places to kill the tumor. So that's also a big challenge. And then finally, it's really this immune system, which we've been thinking about for many decades, for many cancers, as I mentioned. But in brain tumors, this is particularly difficult because the immune system is suppressed, and that's why cancer forms. We also end up suppressing the immune system just because of swelling in the brain. We use steroids to counteract that swelling, and as a consequence, inhibit the immune system from doing its job. Some of the treatments that we use, the radiation and chemo, can also affect the immune system. While it's trying to kill the cancer, can also have a negative impact on the immune system, and that limits our ability to do immunotherapy as well. So these are the challenges that we have to overcome in order to get the bigger benefit from the drugs that we're currently studying in clinical trials and in the future. So over the years, as shown on this slide, we've tried many different types of immunotherapies, from antibodies to EGFR, PDGFR, VEGF, to vaccines, from taking the whole tumor and making a vaccine out of the tumor, or making cells and growing the cells and making a vaccine out of the tumor cells, or more recently, making neoantigen peptides. We've also used viruses to inject into the tumor to kill the tumor and induce an immune response. And more recently with small molecules such as IDO inhibitors or TGF-beta inhibitors. And obviously, and more recently in the last 5-10 years, checkpoint inhibitors, where we're trying to get rid of the brakes on the immune system that cancers tend to cause so that we can get a better immune response against the tumor. And then probably in the last few years, you've heard about CAR T cells. These are taking your own T cells out of your body, engineering them to attack a specific protein which is present on the tumor cells, and then injecting them back into your body to go and do its job. And that also has shown some limited benefit as well. Again, the challenges are that even though we've tried them all, we need complete removal so that we don't need steroids in many of these patients. And we do use steroids around surgery, so that also causes some immunosuppression. And then some of these newer approaches, especially the checkpoint inhibitors or the cellular CAR T cells, can actually create a lot of inflammation in the brain, and that causes symptoms such as seizures, headache, which the only treatment we have is to give steroids and counteract that effect, or Avastin or other drugs that affect angiogenesis. So it's a balancing act of trying to stimulate the immune system to go kill the tumor, but not stimulate it so that it causes side effects in the brain, such as cerebral edema and seizures and headaches and weakness. So this is really what we're trying to do, is find that balance of using a variety of immunotherapy approaches in an intelligent way for each patient to get the effect we want without the side effects. And this has grown exponentially in the last 5 years. Many companies have made drugs targeting, as you can see on here, a variety of molecules on the immune system that can stimulate the T cells to attack the tumor, or prevent the brakes on the tumor from preventing the T cells from killing the tumor. So these are things that are being studied. They were initially studied individually, and now there are more combination studies going on for a variety of cancers, and we're also using these in brain tumors more recently. So the next few slides just talks about the potential to revolutionize treatment for brain tumors. And we've seen this for other tumors, as shown on this slide. And this is an old slide, there are many more approvals of these drugs, which are on the left side. Pembrolizumab, nivolumab were the first set of drugs, and there's other drugs on that list. And you can see they're being studied in all the cancers, which are in the different columns, and many of these diseases that the drugs are being marketed, meaning approved for those diseases. And like I said, this is a 3-year-old slide, and it's probably double or triple the number of approvals in a variety of cancers as single agent checkpoint inhibitors, as well as in combination with other drugs. But even in those diseases, it's not 100% response rate. Melanoma, which is the most immunogenic, the single agent checkpoint inhibitors is about a 20% response rate, and combination is about 40-50% response rate. So even in the best case scenario, we still are a long way of getting a cure for all of our patients. A subset of patients respond really well and are durable responses for many years. And one of the needs is really understanding whose responds, what are the biomarkers, and can we do better? And some of those markers may be relevant to brain tumors, but may not necessarily be so. because the immune system is different in the brain compared to the other organs. And this is just a slide showing the different types of biomarkers that we need to understand from is the tumor releasing antigens? Are the antigens being presented to the immune system properly? Is the immune system getting activated properly in the lymph nodes? Are the immune system cells actually going into the tumor where it needs to in order to do its work? How does the blood-brain barrier determine that? And then once it gets there, are they being inhibited by checkpoints that limit their ability to actually do the job that they're supposed to do? So these are all factors that play a role in why the immune system may not be able to do the job, or why these drugs may not work the way they should in brain tumors that we really need to understand better. We're only beginning to start to understand this process in a way that we can actually interrogate in our patients over time. I will tell you the technology and the tools that we have now are so much better than even 2-3 years ago, that we can really understand what's called the tumor microenvironment. When we take a piece of tissue, look under a slide, what do we see? Besides tumor cells, which is what we've always focused on, what else is there? What are the immune cells? How much immune cells are there? What kind of immune cells are there? Are they active, not active? And what's preventing them from doing their job? And we can have the tools now where we can actually do this on a single patient level. And that's really what's needed, I think, to make progress in this disease. One of the other markers are what's called tumor mutation load. And this slide just shows all the different types of cancers from the Cancer Genome Atlas. And you can see melanomas on the right side, they have the highest tumor mutational load. And that's probably why they respond so well. And then if you look down the list going to the left, you'll see glioblastoma is a lower mutational tumor load. And so that's maybe why it doesn't respond as well. And then keep going to the left, you'll see low-grade glioma, and that is even lower tumor mutation load. So this may be one of the reasons why immunotherapy doesn't work as well in brain tumors as opposed to melanoma. We do know that this tumor mutation load has an important association with clinical benefit, and this is for lung cancer, but like I mentioned, for brain tumors, we don't see as good response. But even in lung cancer, you can see that patients who have high tumor mutation burden, the red here, do a lot better in terms of progression-free survival on the left side, as well as overall survival on the right side. So the amount of mutations that are present in a tumor cell dictate how strong an immune response, how the immune response can react to that tumor and help prevent that tumor from growing. So this is an important marker, a biomarker of response to checkpoint inhibitors. And for brain cancer, like as I mentioned, we always worry about inflammation in the brain. Here's an example in the literature of a melanoma patient who received checkpoint inhibitors. And you can see the lesions in the lung on the left, several lesions that disappeared on this after treatment with the checkpoint inhibitors. In the brain, there was an asymptomatic lesion in the left temporal lobe, as shown on the bottom left brain MRI, and you can see that also disappeared. And the patient didn't have any cerebral edema, didn't have any side effects from that treatment. So the point here is that checkpoint inhibitors can work in the brain in certain contexts without side effects. And there's many other studies showing this for metastasis from other cancers to the brain. Checkpoint inhibitors can actually have a significant response in the brain without undue toxicity in those cancers. And here's a summary of that data that was shown in several different studies showing a response rate, intracranial response rate of over 55% in Checkmate 204, and then in an Australian study of 42% response rate, which is very significant in patients. And what this means that we don't have to do radiation to the brain, we don't have to do surgery to the brain for these lesions. So this is sort of a landmark study showing that checkpoint inhibitors in the brain in certain diseases can have a significant effect. Now, what about brain tumors, other types of brain tumors? I will show this example of a patient of mine many years ago with a unique type of brain tumor called primary CNS lymphoma, who I've treated for almost 10 years with a variety of drugs, and the tumor kept coming back, and we, had transplant, we did a whole bunch of chemos. We ended up doing whole brain radiation in 2015, and even a month after that, the patient progressed. And this was in the early days of lymphoma immunotherapy, where nivolumab was not approved, but we had no choice, so we got it as a compassionate use, the drug. And you can see here before immunotherapy, you see the patient had probably 10 lesions, and you can see 2 of them with the arrows here. And then a couple of months later, the patient responded, and over time, the patient continued to respond many years with just nivolumab. So even in the brain with other besides metastasis, primary brain tumors such as CNS lymphoma can respond. And then there was a study published out of University of Toronto, Hospital for Sick Kids, where they treated 2 siblings who had a genetic disorder a genetic disorder that creates a lot of mutations in the tumor, and those mutations are recognized by the immune system. So so just looking at the right side here, you'll see this blue line looking at the number of mutation these 2 siblings had in their tumor was very high, even compared to colon cancer, lung cancer, melanoma, which we tend to think of having a lot of mutations. When they treated these 2 patients with nivolumab, they had inflammation in the brain after a few weeks, more swelling as shown in the white spots here. But then eventually the inflammation subsided and the tumor actually shrank over time, which is shown here. So here, the tumor came back, and then the tumor regressed after 3 months of nivolumab treatment. So even in gliomas, the tumors that we deal with and that are the most difficult, in certain contexts, in this context, it was a genetic disorder that created a lot of mutations in the tumor and that caused the tumor. Nivolumab had a clinical benefit of regressing the tumor over time. There was some transient inflammation and swelling, and the investigators controlled that with either steroids or Avastin, and then eventually the tumor shrank over time. So it's a proof of concept that we can make a difference there. So to end with, those are just specific examples, specific types of brain tumors, whether it's from metastasis to lymphoma in the brain to gliomas. These are all different tumor types. One of the things that we need to understand is this heterogeneity. What is the immune system in each person, each tumor type, what's called the tumor microenvironment. And I think now, compared to even a few years ago, we have the tools with these new tissue-based analytic tools, such as Immunoscore or NanoString and other tools, where we can actually understand at a single cell level what are the markers in every single cell that we're looking at under the slides, and how does that impact immune response. And I think what we'll see in the future is using that type of big data and doing smarter studies based on the biomarkers that are present in each person's tumor. For instance, if you have a drug that targets PD-1 and the tumor doesn't express PD-1, it's probably not going to be that helpful, and so on and so forth. There's probably about 10 to 20 markers, immune markers in these tumors that we can target right now. And we just need to understand whether those markers are present or not, and which drug or combination of drugs are going to make a difference for these patients. And that's what I think we're all moving towards in terms of personalized medicine and personalized immunotherapy down the line. So I'm going to stop there, Al. I can keep talking forever about immunotherapy, but that's kind of a broad overview of things. And obviously, compared to even a few years ago, we only had PD-1, CTLA-4, and now there's a whole host of new drugs out there. And ultimately, I think the kinase inhibitors are still important. And I think maybe even chemotherapy and radiation are important, because there's examples where radiosurgery or radiation with immunotherapy may benefit some patients, or targeting the kinase inhibitors that's creating an immunosuppressive environment may actually help immunotherapy as well. So I think that's really the future, and we just need to do a better job of doing better studies.
Okay, that was very good. Thank you. There's a bunch of questions. The first one is, we want to learn more about your CAR T cell trial. That's, can you talk about that a little bit?
Yeah, many of these studies are actually, we don't have an active trial, but we are working with others. But the published data is really out of UPenn and City of Hope, and UCSF and other people are doing it as well. And they've targeted like the mutations, like for instance EGFR V3, which is a common mutation in glioblastoma, and or a common antigen IL-13, for instance, in glioblastoma as well. So you identify a common marker that's expressed in the tumor cells, but not in the in the normal body, and a CAR T is going to go and attack those tumor cells. Those initial studies shown that it's feasible. There is some toxicity, as I mentioned, in terms of swelling that needs to be uh, controlled, and we've actually done a better job of how to manage it as we're treating patients. And some of the data suggests that it's doing something, meaning it's targeting those cells. We can see a imaging effect of killing of tumor cells in some cases, and mainly case reports or small percentage of patients in a trial that seem to actually benefit. But it's not a big benefit yet. And again, it probably gets back to what's on the slide, the tumor heterogeneity. Even a patient that's EGFR V3 positive, it's only a small percentage of patient cells in the tumor that are positive. So say if the CAR T is successful, gets rid of 100% of the EGFR V3 cells, the other cells are still there, and if they don't stop growing, they'll still continue to grow the tumor.
Would it make sense to, do you remember ICT 107, where they took, it was 5 or 6 different targets and combined them to make a vaccine against those 5 or 6 targets just for that reason? Maybe do the
Exactly.
CAR T cell exactly the same.
Exactly. So the initial wave of vaccine therapies was just one antigen, including EGFR V3. There was a vaccine for that too. And then we got smarter and did multiple antigens, as 6, 10, etc. And then, we also did tumor lysates, where we exposed all the antigens present in the individual person's tumor to the immune system as well. And all of those have shown some benefit, but not in a big way. And in the CAR T field, yeah, you can go after multiple antigens as well. It's just obviously the cost and the logistics gets more complicated. But that's probably either you find an antigen that's universally expressed, that's unique to glioblastoma, that's present in every single cell, or you have to do multiple CAR Ts targeting a variety of cells so that you cover all the cells that are present there.
Okay. What clinical trials are you involved with right now?
So we're involved with a variety of clinical trials using many of the immunotherapy drugs that we just talked about, checkpoint inhibitors, as well as biomarker-driven trials, mainly on the kinase inhibitor side. For instance, EGFR amplified or mutated patients, we have a trial of a pulse dosing of a kinase inhibitor. We are putting together trials of where we combine checkpoint inhibitors with kinase inhibitors as well. As I mentioned, some of the kinase mutations in the tumors drive the immunosuppressive microenvironment. So we want to do both combinations to try to get a better response. We're also doing trials of devices and infusions of drug directly into the tumor, where for instance, a company called Medicina, where we inject through a convection enhanced delivery drugs that deliver a toxic payload to a tumor expressing IL-4 receptor, which is highly expressed in glioblastoma, for instance. So we're
How
looking
is
at,
that working out? Do we have
well,
any early results?
well, we had presented at ASCO and SNO last year that the data looks very good. In fact, I can show you some slides because they're in the slide deck. So normally, this IL-4 expression is actually receptor expression actually predicts poor survival. So the red here is high expression; they do worse. So when we in the trial summary data, the patients, I don't have it here, but the patients who actually had the high expression actually did the best. So it actually reversed the prognostic value of that biomarker, just like in breast cancer. In the old days, breast cancer, if you're HER2 positive, the survival was very poor. And then when we made a drug to target HER2, like Herceptin, HER2 positive patients do a lot better than HER2 negative patients because we can actually target that mutation.
With this slide, that's just one dose at the beginning?
Yes. So these patients are, it's a surgical trial, right? It's a convection enhanced delivery. So the patients get an infusion based on the tumor and according to a volume with convection enhanced delivery to the tumor. But one treatment, yes.
Okay. Getting back to delivering these drugs, we understand the problem about the blood-brain barrier where the drugs can't get in. So you're saying convection enhanced delivery is one way of getting around it, but it's a surgical procedure that you really can't easily repeat over and over. What other ways can we get around the blood-brain barrier problem? Anything you're working on?
So there's, first of all, the convection and delivery, we've actually been able to do it compassionate use on repeatedly in patients,
Okay.
in a couple of cases. So you could do it again, actually, if the patient responded initially. Uh, the the other way is making drugs that get across the blood-brain barrier actually get across with new technologies. And we worked with a company called Angiochem that had a peptide bound to, for instance, this was Taxol, an old breast cancer drug, and putting that peptide that binds to the blood-brain barrier and actually gets the chemo across the blood-brain barrier. Uh, uh, Actually, we saw responses in patients with gliomas as well as in leptomeningeal cancers from breast cancer. And there's a larger study, randomized study, that we're waiting for it to start in the near future. The other way to do it is, there's several other ways to do it. One is to disrupt the blood-brain barrier. And people have done various approaches, including intra-arterial blood-brain barrier disruption. And then more recently, I think people may be familiar with this, is focused ultrasound. So it's an external device that uses ultrasound waves to disrupt the blood-brain barrier, which we think is disrupted already, but not very efficient. But this allows even more drug to get into the tumor and to the infiltrating component of the tumor. And maybe the drugs that we're using right now, they're good drugs, but they just don't get in very well. And if you increase the delivery of the drug into the tumor, you could get a bigger response. And some of the studies with several of the ultrasound companies showed some benefit in some patients that have been treated and reported in the literature. So I think that may help broadly across the field. And there are many companies now doing ultrasound-based technologies to increase the delivery of drugs to the brain.
Okay. What are your thoughts on using Avastin for glioblastoma? There was conflicting trials. What is the current thinking right now?
I think Avastin is a benefit to patients with recurrent glioblastoma. And I think there's a subset of patients, probably about a third of patients, that I really think it really benefits quite a bit. And we kind of know who those patients are. Those are patients who have a lot of swelling, need high dose steroids, and on imaging you can see that swelling, and you get that flavor of radiation type of necrosis look to the imaging. And I think those patients are probably the ones that benefit the most from Avastin, mainly symptomatically, getting rid of the steroids, which is immunosuppressive. So that I think that's a subset that really respond really well. So I think it is a valuable tool, even though upfront it may not have had the benefit we're all looking for. A subset of patients really, I think, benefit quite a bit. And
Is it?
then,
Oh, good.
and
Sorry.
then there's more data nowadays in terms of checkpoint inhibitors that Avastin may also help, for instance, control the negative inflammation from the checkpoint inhibitors and allowing us to avoid steroids so that the checkpoints do their job and the immune system does its job without negating it with having to start steroids.
Okay. Is it relatively safe to use Avastin on pediatric patients? Like, I worry that you could hurt growth. Is that true?
That I
Of the rest of the body, I'm talking about.
Yeah, right. That's a very good question. I don't know the data on that. I know my pediatric colleagues have used Avastin and use it.
Well, you got to weigh the risk versus the benefits, obviously.
Yeah, the risk of it, and as you know, radiation and chemo stunt growth in a huge way. I think the Avastin would be much more negligible than what we do already.
Okay. We talk about steroid use a lot. Do you remember the drug Zaricept? Well, that might be before your time. Oh,
No, no,
was it?
no. That was yeah. No, when I was a fellow, we were doing the CRF, corticotropin releasing factor. Yes.
Right. Back then, it kind of failed because it just greatly reduced the use of steroids, but didn't eliminate it in enough people to make it worthwhile. But I'm thinking now that you have all these immunotherapies that are sensitive to steroid use, that might be a benefit, and it should probably be tried again. What do you think?
I think that's a very good idea, and there are also other drugs like that that may be a benefit. You're absolutely right.
Okay. Let me see. Do you see any drugs in the pipeline that might make a big impact on the glioblastomas?
Well, I think there's a lot of promising drugs out there, early, preclinical and then early stage clinical that um, have really promising data that could be very impactful. Obviously, many things are promising in the preclinical level, and they may or may not help later. But I think, yeah, there are many drugs out there that I think are promising. Um,
What would your number one or two pick be?
well, I think so. There's one drug, I think, a PI3 kinase inhibitor that was originally with Genentech, and it's now with a small biotech that have reported some promising results. I think it's unpublished though; it's just abstracts. And I think it's Kesari Therapeutics. Um, and then, yeah.
Okay. Would it be fair to say that most hope would be on combinations of therapies rather than an individual therapy?
Absolutely. I think it's clear that combination is the best approach, whether it's combination kinase inhibitors or with chemo or with immunotherapy, that is the best approach. But, studying it in with a rare disease like brain tumors are is difficult and challenging. And we have done combination therapies in the past, and the side effects have to be managed better too. So from the kinase literature, we've learned that when we have done combination, the toxicities are more. We end up reducing the dose of drugs so that there may not be enough that gets into the brain. So we either have to develop better drugs that get into the brain better at low doses, or we have to do alternative like pulse dosing and things like that in order to be able to deliver enough drug in each of these patients and make it tolerable.
Okay. We've been seeing a lot of phase 3 trials fail where the phase 2 trials look fantastic, like the Celldex trial, for example, and even ICT 107. How could these things happen? Why do they all of a sudden fail? Any ideas?
By nature, phase one study means you just want to find out what the safest dose is, right?
Right.
Phase two study is there enough of a signal on its without any comparator arm, enough of a signal enough response, whether it's objective response, progression-free survival, to make it worth it to do a randomized phase 2 or phase 3 study where you're comparing to some other standard of care. So by design, phase 2 is just designed to say, hey, this looks promising enough that we should then do the bigger study. But it doesn't guarantee that it's going to work. And that's what happens in the phase 3, that, you know, it just doesn't give enough of a signal to make it better than the standard of care.
So, for example, like the Celldex vaccine, it did good in the phase 1 trials. It did very good in multiple phase 2 trials. And then in the phase 3 trial, it actually did good, but the control arm did even better.
Yeah.
So which do you believe? Do you believe that it might even have some use? Because, as you said, by itself, one of these drugs can never really have a major impact because that one was targeting EGFRV3.
Right.
But what about using that drug as part of a combination? So you get rid of the EGFRV3 component and then use something else to knock down 2 other components.
Yeah, no, you know, in the ideal world, I would love to combine about 10 different drugs from 10 different companies and do it all at once, right? If it's safely possible. But, you know, it's very difficult to even combine 2 drugs from 2 companies, much less trying to combine, you know, with more than 2. So I think that one of the challenges is how can we do a study where we do a combination of checkpoints, vaccines, kinase inhibitor, et cetera, together, that we think is going to really be the home run. But that's a challenging study to do for a variety of reasons, including getting the companies on board to want to do that. Everyone's always worried about side effects from the individual drug, as well as just combining with one other drug, much less doing a whole combination of things all at once. But I think it's something that I know outside the US, Europe, they tend to do a lot of combinations like that, much more easily, vaccines and peptides and viruses. But here it's very difficult to get. I think it's aspirational. We want to do that, but it's not easy.
Okay, it's funny this came up because I'm working on the solution to that problem. I've been working on it for 24 years right now, but
Right.
finally last week we had a bill introduced into Congress. I don't know if you heard of this yet. It's the Promising Pathway Act. Have you ever heard of that?
No.
This is wild. Uh, One of my ideas has always been drugs like the Celldex drug are very, very useful, but not by themselves. They'll never have a major impact by themselves because they're hitting one target. So the key would be to use it in combination with other treatments, but you can't because it's no longer available. The only way you're going to actually get access to these drugs, the same with like ICT-107 and even like MDNA 55, what you're talking about, is to get them approved sooner. So this bill would actually change the order of research. Instead of like phase 1, phase 2, phase 3 approval, it would be like phase 1, small phase 2, conditional approval, and then more research using a registry to track every single patient who uses it, see which drugs they use, the combinations, and the outcomes. And then eventually, if they hit a certain target, they'll get full approval. Or if they don't do well, they just get removed. Um, and this is actually in Congress right now. We're working on a plan to push it through. It sounds crazy. And when I first heard about it, I said it's insane. But if you look through it and you look at all these combinations that we have to do, one of the other problems with combinations is you would have an idea for 10 different combinations. You'll do them. You'll do 3 patients with each combination, and it's meaningless, even if you have success. Under this plan, every doctor in the United States who has ideas comes up with their own combinations. The outcomes will be tracked, and you would be able to see the research and say, oh, 200 people did this, and they had double the survival of comparable groups. Let's work on variations of this combination. Well, this combination had too much toxicity, forget about that. You wouldn't be doomed to repeat the same mistakes everybody else makes with toxicity. And you get your creativity back, just like the times of Dana-Farber back with leukemia in the old days, if you remember that.
Yeah. Well,
So
this is a great idea. Please send me that link to that act. But it also reminds me of, I guess, the Right to Try Act. Right? I don't know whether
this is different.
I know it is different because you get to. Yeah, I understand it's different.
The Right to Try is good. We actually have a program where we help people get access to Right to Try drugs right now. We have a few people doing vaccines under the right to try. The trouble is it's really expensive.
Yeah.
Under this new law, uh, an insurance company will not be able to deny payment for it based on it being experimental anymore. They don't have to pay, like with Optune, it took them 10 years to start paying for it. But at least they can't just say it's experimental, so we're not paying for it. So there's a good chance most of the insurance companies will pay for it. Most likely the drugs will be cheaper. It'll cut off about 95% of the cost of developing a drug, uh, and probably 7 years or so. Uh, Immediately we should get 5 to 10 new drugs. We could start doing combinations, and every year you get a couple more drugs, so that we have a lot of tools in the toolbox to try to put together with people like you, um, to really hone in on the ultimate cocktail. And I think
No,
that's where we're going to make the biggest change we ever had in the brain tumor field.
no, absolutely. And Al, as you know, I do a lot of off-label treatments, right? When we run out of clinical trial options. So this would allow, we spend a lot of effort and time trying to get a drug approved by companies. And some of the companies have been very kind. They have access programs so that even when the insurance denies, patients can get access through various programs for those drugs. And I think this sounds like even another version helps us get these drug access even faster and easier for these patients, which
Right.
is
Once they're approved, it goes faster. We also have a program where we help people get access to off-label drugs or expanded access drugs or all that type of stuff too. And we also have a registry where we could follow all of these cases. So if you're doing a unique combination, instead of setting up the infrastructure to do an actual small phase 1 trial, you just run through the registry. Once you get whatever number of patients you want, it could be analyzed, especially if they're off-label, where you don't have to worry about FDA stuff. And that's the fastest way. And we'll use this system if this Promising Pathway Act ever gets approved to track these patients.
this is great.
The Promising Pathway Act requires all the patients to be within the registry. As a matter of fact, there's a large penalty if not more than 90% of the patients participate. So it's an exciting thing. We're going to be sending out a lot of information in the next week or two about how people can help get this approved. I
Yeah,
think I think
I'd
it's going to make a big difference.
love to participate, Al. It sounds great.
Okay. Well, that's about it for our webinar. We don't have any more scheduled. If something important comes up, we'll let you guys know and set up another webinar. But for now, have a happy Father's Day, and thank you, Santosh. It was
Thanks,
very
Al.
interesting.
Okay.
Bye-bye.
Bye-bye.